Dear students, greetings of the day. I am Dr. Sujata from Sushant University, and today in this lecture series, we will learn about a basic introduction to research. So today in this lecture series, the key highlights are need for research in the pharmaceutical sector, importance of experimental design, ethical considerations in research with concern to plagiarism, link between research, design of experiments, and ethical conduct in pharmaceuticals. So first of all, why there is a need for research? Why research is crucial in pharmaceuticals? First of all, research is important due to innovation and drug development. The pharmaceutical market, it is flooded with a range of drug products in different categories like analgesics, antipyretics, NSAIDs, antiviral drugs, anti-neoplastic drugs, and these all are outcomes of rigorous research. Second is in improving patient health. So it's a common philosophy that when we are looking for a cure of any particular disease, first of all, we need to look for the cause of that disease, that what is the underlying cause of this disease, because when the cause is known to us, then only then we can look for a better treatment strategy of that disease. So in this way also research is important, that what will be the cause of the disease, what are the pathophysiology of the disease, what are the different factors that are responsible for the causing of this particular disease. Third, to meet the regulatory standards of the different organizations like FDA and EMA regulatory, these research is important. If we're taking example from the pharmaceutical sector, so in the development of COVID-19 vaccines, it is a result of the accelerated research effects. Also, the insulin and other medications are developed through years of research and clinical trials. Next, come to the need for design of experiments. So why designing of experiments are important? It helps in identifying and clarifying the objectives of research, and it ensures that the collected data is relevant and scientifically valid, that is data integrity. And it also reduces the cost, time, resources, and spent on the ineffective experiments. Next, coming to the experimental design techniques, what are the different experimental design techniques? So generally 3 different techniques are there. First one is the randomized controlled trials. They're widely used to test drug efficacy. For example, testing the efficacy of a new antibiotic using a double-blind RCT to compare the existing treatments. Second design is the factorial design. So factorial design is applicable when we look for different variables. For example, where if you are giving different doses of paracetamol to same patient, then 2 different doses, that is one variable. Then secondly, we are giving a different dose of paracetamol to the patient, like given after meals and before meals. This before meal and after meal, this is another variable. So when we are considering more than 2 variables in any of the experiment, then we are using the factorial design. Third design is the crossover design. Next, coming to the type of experimental designs. So generally when a new drug is discovered, then it must undergo these type of experimental design like the preclinical studies, clinical trials, and the observation studies. So what is there in the preclinical studies? Basically, the preclinical study, it is of 3 types: in vitro studies on animal subjects and in silico, ex silico studies. So in vitro studies, we are doing the studies on the cell lines or the tissue cultures, etc., seeing the safety and efficacy or the toxicological studies of any drug. And in animal models, we are using the animals like rats, rice, rabbits, guinea pigs, etc., to check the safety and efficacy of any drug. In ex silico studies, we are using softwares like CAD, QSCR, and computational screening techniques, etc., to look for the safety and efficacy of any drug. Next, coming to the clinical trials. In clinical trials, we have 4 different phases, that is phase 1, phase 2, phase 3, and phase 4. In phase 1, it is generally to look for the safety and dosage of any particular drug. And thus, we are taking about About 100 subjects are there. Healthy human volunteers are there in phase 1 studies, and phase 2 studies, it is generally for the efficacy, to check the efficacy and side effect of the drug. So about 100 to 300 human volunteers are there in phase 2 studies. In phase 3 studies, phase 3 study generally aims to look for the efficacy and monitoring of the adverse reactions of the drugs, and it is generally over a large number of human volunteers, about 3,000. After undergoing all these 3 phases, the drug enters into the stage of post-marketing surveillance, from where if any drug, after launching into the market, if the general population is facing any problem related to that particular drug, then the drug is withdrawn from the market shelves. For example, phase 3 clinical trial for the Pfizer biotech for COVID-19 vaccine tested that the drug in thousands of patients to assess the efficacy and safety. Another example of this is rofecoxib and rosiglitazone. These are 2 major drugs which are withdrawn from the market due to their side effect, that is, they're mainly causing cardiac problems in the general population, so they are withdrawn from the market. Next we will learn about the ethics in research. So when we will talking about the ethics in research, one major word that comes in our mind is plagiarism. So what plagiarism exactly is? Plagiarism is just using someone else's work. So copy paste of data is not allowed for in research studies. I'll give you an example from the pharmaceutical sectors. Once in the Vivox scandal is there related to the drug, that is rofecoxib, in which improper data handling lead to reporting of major legal battles for Merck. As this particular drug, it causes increased risk of heart attacks in the patient, but due to improper data, presentation of the data, all these consequences led to the withdrawal of this particular drug from the market in 2004. So plagiarism, it basically leads to scientific fraud, legal and financial ramifications, and loss of public trust. One more example is in 2012, Seralini study on genetically modified corn and its link to cancer was later criticized for scientific misconduct. Next, so how we will prevent plagiarism in pharmaceutical research? As I've already told you that to remove the plagiarism from our research work, certain softwares are there. So which gives a very clear indication that what is the level of plagiarism in our reporting of research work. So these softwares are Turnitin, Dilbert, Urkund, etc. We can take help of these softwares to remove plagiarism. Next, coming to the ethical conduct in pharmaceuticals. So what is ethics all about? First of all, we should respect the persons. For example, in our previous slides, we have discussed about the different clinical trials in which we are using the human volunteers. So there will be an informed consent and autonomy of all the human volunteers, which is very much important. Second is we must meet the regulatory requirements, that is the GMP and GLP requirement, the good manufacturing practices and good laboratory practices. All the drugs are manufactured in accordance with all these standards and specifications. When we are talking about the real world implications, then protection of human rights and avoidance of exploitation and ensuring patient safety is of importance and of major concern. Students, in this lecture, we learn about the need for research, different type of design of experiments, need for design of experiments, and ethics in pharmaceuticals. Thank you.